Today, we would like to discuss single-nuclei sequencing and NASH liver disease, in title of Resolution is the Revolution. By unmasking the cellular actors of environmental toxicity, from zonated HSCs to exhausted T cells, we define the roadmap for next-generation liver therapeutics. The invisible is now visible. From bulk averages to cellular atlases, bulk sequencing masks heterogeneity by providing average expression data. Single-nuclei sequencing, SNSeq, unlocks frozen biobanks, capturing fragile adipocytes and hepatocytes, often lost in enzymatic digestion. In TCDD toxicity studies, SNSeq successfully recovered 11 distinct cell types from frozen tissue, providing a high-resolution map of hepatic injury.
The resolution revolution distinguishes the smoothie of bulk RNA-seq, which averages gene expression and masks rare populations, from the fruit salad of SNSeq. SNSeq profiles frozen archives without digestion stress, revealing high-fidelity cellular landscapes and critical spatial nuances necessary for precision medicine.
The geography of injury defines portal versus central zones. In the liver lobule, Zone 1 near the portal vein is oxygen-rich, hosting PAT-HSC fibroblasts. Conversely, Zone 3 near the central vein acts as a hypoxic metabolic hub with high CYP1A2 activity, making it the primary site for toxicological damage and metabolic stress. Profiling the architects of fibrosis, portal-associated HSCs, PA-HSCs, are homeostatic bystanders marked by NGFR and MAFA4. Central-associated HSCs, CA-HSCs, located near the central vein and marked by ADAM-TSL2 and LPAR1, are the primary pathogenic collagen sources. Our therapeutic strategy focuses on targeting these ADAM-TSL2 positive CA-HSCs via LPAR1 inhibition. The emergence of NAFLD-associated macrophages, NAMs. This signature, characterized by Trem2, CD9, and Gpnmb, is absent in healthy livers, but expands to over 20% of the macrophage pool during injury. NAMs are highly conserved and function as scavenger receptors for clearing apoptotic cells, marking a critical transition in immune pathology. The double hit, diet plus microplastics. High-fat diets combined with microplastic intake accelerate disease progression. This synergy increases inflammatory S100A6+ macrophages, outcome B, driving angiogenesis and tumorigenesis, while decreasing protective VSIG4+ macrophages, outcome A, essential for tissue repair, leading to severe inflammation and fibrogenesis. Conserved biology across species. Our research confirms that mouse models provide a valid guide for human territory. The NAM signature, Trem2/Gpnmb, HSC zonation, portal NGFR/CNGFR, central ADAM-TAS-SL2, and vascular injury patterns are highly conserved, validating the mouse map as a reliable blueprint for human hepatic pathophysiology. The language of the liver, intercellular crosstalk. Connectonomic analysis reveals a complex network of interactions between HSCs, macrophages, endothelial cells, and T cells. Key pathways include HSC secretions, vasoactive signaling via endothelin, and specialized macrophage to T cell communications that coordinate the organ-wide response to environmental stressors. From map to medicine, precision targets. We identify 3 pillars. 1. Target the villain by inhibiting LPAR1 plus CHSCs to stop pathogenic collagen. 2. Modulate immunity by restoring the balance between VSIG4 plus repair and S100A6 plus inflammatory macrophages. 3. Restore vasculature by protecting c-KIT plus LSECs, ensuring comprehensive organ recovery. The pathogenic origin of sentrilobular fibrosis. Single-cell analysis reveals that LPAR1 is specifically enriched on pathogenic Ca-HSCs during injury. Targeting LPAR1 specifically inhibits fibrosis by acting on this localized subpopulation, proving that spatial precision outperforms blunt antifibrotic approaches by preserving quiescent Pa-HSCs and liver homeostasis. The multiple hit hypothesis details. Microplastics are not inert. They synergize with high-fat diets to significantly increase NAFLD activity scores beyond diet alone. Mechanisms involve altering the immune microenvironment, increasing inflammatory S100A6+ macrophages, influencing hepatocyte stemness, and promoting CD4+ recruitment alongside CD8+ T-cell exhaustion. Cellular profile, the NAM. Originating from differentiated monocytes or Kupffer cell subtypes, NAMs are highly active and pathogenic markers of steatohepatitis. Enriched for endocytosis and lysosomal degradation genes, they play a critical role in clearing lipid-laden apoptotic cells, primarily identified by Trem2, Gpnmb, and CD9 markers. Macrophage plasticity, toxicity versus pollution. TCDD toxicity triggers a 24.7% expansion of Gpnmb plus clusters, mirroring NAM patterns. Simultaneously, microplastics shift the polarization balance away from protective Vsig4+ tissue repair macrophages toward pro-inflammatory and angiogenic S100A6+ populations, fundamentally reconfiguring the hepatic immune landscape. Adaptive immunity, T-cell exhaustion and dysfunction. Chronic activation triggered by microplastics and high-fat diets leads to a transition from naive CD8+ T cells to an exhausted state, CD8TX. Molecular markers like LAG3 and PD1 are upregulated, resulting in a failure of immune surveillance and a shift toward a pro-inflammatory, tumorigenic environment. Mechanisms of toxicity, pathway reprogramming. Toxicity involves a coordinated transcriptional shift. Parenchymal hepatocytes undergo a massive metabolic shutdown of lipid, cholesterol, and amino acid metabolism. Simultaneously, non-parenchymal cells like stellate and endothelial cells upregulate injury responses, including RAS signaling, inflammatory pathways, and fibrotic signaling cascades. Transcriptomic conservation validated. Mouse cRNA-seq findings are not artifacts. They provide a reliable blueprint for human hepatic pathophysiology. The ANM signature, Trem2 Gpnmb, correlates directly with human fibrosis stages, and HSC zonation patterns, NGFR plus portal versus 8M-TSL2 central, are identical across both species. Unlocking the archives. Traditional scRNA-seq requires fresh tissue, limiting retrospective research. The breakthrough of single-nuclei sequencing, SN-seq, on frozen tissue yields cell-type-specific transcriptomes indistinguishable from fresh methods, allowing us to retrospectively map cellular landscapes from massive historical clinical biobanks. Deconstructing the organ cell by cell. Moving from bulk averages to single-cell resolution allows us to profile over 43,000 individual transcriptomes. This granularity is essential to resolve tissue heterogeneity, map spatial zones, and identify rare pathogenic populations that are invisible to traditional bulk sequencing methods. The myth of cellular homogeneity. Single-cell transcriptomics debunks the old dogma of liver uniformity. We now recognize that hepatic stellate cells, HSCs, are topographically distinct. Blue-coded PA-HSCs are portal vein associated, while red-coded CHSCs are central vein associated, each performing fundamentally different roles in health and disease. Unmasking the master builder of scar tissue. In centrilobular injury, specific ADAM-TSL2+ CAHSCs drive pathogenic collagen deposition. These cells are the dominant architects of liver stiffening and fibrosis, localized specifically around the central vein, as visualized in high-resolution immunofluorescence imaging. From zonation to precision therapy, understanding spatial logic reveals LPAR1, lysopacitic acid receptor 1, as a specific therapeutic target, expressed specifically on pathogenic KHSCs. Pharmacological blockade of LPAR1 inhibits fibrosis without disrupting the liver's homeostatic functions or other cell populations. The macrophage plasticity switch. Microplastics force a dangerous shift in the immune landscape. This involves the depletion of restorative V-Sig4+ Kupffer cells and the recruitment of aggressive S100A6+ macrophages that drive angiogenesis and tumor signaling, tipping the balance from repair toward chronic inflammation. The rise of the NAM. Chemical toxicity via TCDD triggers massive immune infiltration, mirroring severe NASH pathology. This universal response involves a 24.7% expansion of the Gpnmb plus, Trem2 plus, CD9 plus phenotype, representing a conserved cellular reaction to toxicological injury across various diet and chemical models. A mirror to human pathology. The cellular shifts observed in mouse models are highly conserved in humans. NASH patients exhibit the exact same cellular shifts and molecular markers, including zonation markers, MFAP4, RGS5, and the NASH signature, Trem2, Gpnmb, which correlates directly with clinical fibrosis scores. The new model of environmental toxicity. Liver disease is an environmental cellular cascade. Convergent pathways from HFD, microplastics, and TCDD trigger zonal injury at the central vein. This induces an immune shift to NAMs, activating KHSC architects to drive fibrosis, eventually progressing to a fibrotic liver and hepatocellular carcinoma. Precision targets for next-generation therapy. High-resolution understanding enables precision medicine. 1. Target the source by reducing exposure and modifying diet. 2. Target the architect using LPAR1 inhibitors for KHSCs. 3. Target the immune system by modulating NAMs and restoring V-Sig4. We can now target pathogenic nodes without harming the healthy organ. Thanks for your attention.